has influenced the evolution of the three life domains. So actually I will be talking about how horizontal gene transfer has importantly impacted the evolution of one particular domain of life. This is the Archaea, and David will talk later about endosymbiotic gene transfer and how this has influenced the evolution of photosynthetic eukaryotes. So I'm going to concentrate now on prokaryotes. And this is an example of work that we have been doing in our lab using a metagenomic approach, and we have identified an important level of horizontal gene transfer in the pangenomes, that is in the collective genomes of particular lineages of, in this case, uncultured marine Archaea, and which are the, what I will comment about the implications that these observations have on the evolution of the whole archaeal domain. So to start with, because I'm not sure if you are familiar with the Archaea, just a very general overview on what they are. Archaea, the third domain of life, was discovered by the end of the 1970s, and at that time they were discovered based on species that were isolated and maintained in culture, and they were divided in two major groups: the Crenarchaeota, all of which were hypothermophilic species living in deep seabeds or in hot springs at more than 80 degrees Celsius in temperature, and the Euryarchaeota that encompassed a much larger variety of phenotypes with hypothermophilic organisms here in red at the base of the Euryarchaeotal branch, other thermophilic organisms growing at intermediate temperatures, and then the halophytes, which were called at the beginning halobacteria before it was known that these were Archaea, and also several lineages of methanogens that were mesophilic, living at temperatures like you and me. So this was at the beginning, but when the first studies, environmental studies using molecular tools to describe the diversity of Archaea in the environment were applied, people like Jed Fuhrman, Ed Delon, and many others began to discover and to determine 16S ribosomal RNA gene sequences in environments such as the sea that occupy this part, the defined groups within the Archaea, both in the Crenarchaeota and in the Euryarchaeota branches. They call these groups Group 1, and then groups 2 and 3 for these two Euryarchaeota groups that were planktonic Archaea. And especially this group 1 attracted the attention of researchers because these were planktonic Archaea, even if they were not in culture, but meaning that these were mesophilic Archaea, and this was an exception to the rule until now known, because all Crenarchaeota known so far were hypothermophiles. So ferberin, this group was also quite abundant and diverse in oceans, and it began to be discovered also in other environments, so that people tried to isolate them very hard. And it took more than 10 years to isolate the first member of this group, who is called Nitrosopumilus maritimus, and it turned out to be a chemolitho-autotrophic aerobic ammonia oxidizer. So it gained energy by the oxidation of ammonia with oxygen, and it was an autotroph. It was also able to fix carbon. Today, this group 1 of or mesophilic Crenarchaeota is known as a different phylum, the Tomarchaeota, so they have been renamed, and together with two other groups of hypothermophilic organisms, the Igarchaeota and the Purarchaeota, they form a monophyletic group that is called the Tag superphylum. Okay, this is taxonomy words, but you may hear about that in general in literature. So what do we know about this group 1 Archaea or Tomarchaeota? So we know that they are highly diverse, not only in the sea, in oceans, but also in soils and freshwater systems. Most of the lineages are non-cultivated, so there are only few species in culture, and none coming from the deep sea. But we know that they are most abundant in the deep sea, so they can make up to between 10 and up to 30% of phytoplankton, of planktonic prokaryotes in general, and small eukaryotes. So they are relatively abundant in the deep sea, and so they are important for the ecosystem, the general marine ecosystem. And, but they are also present in other environments, and at the beginning people thought that they were only mesophilic species living at relatively mild temperatures, but actually they have been also discovered in hot springs. So you have here these two groups correspond to thermophilic organisms. So the idea now is that the whole group adapted secondarily to mesophily, to low temperatures, if you want, from a hydrothermophilic ancestor. Okay. Regarding the metabolism, we think that many, if not most of them, gain energy by oxidizing ammonia with oxygen to produce nitrite, and because of this activity, they are very important players in the nitrogen cycle, even more important than bacteria. So they have really raised a lot of discussion in the scientific literature because of their implication in the nitrogen cycle, and they are also very important for the carbon cycle because at least several of them are autotrophic, so they are able to fix CO2 inorganic carbon by using a variant of this hydroxypropylene hydroxybutyrate pathway that is typical for nitrosofumidos maritimos, for instance. Many of them may be also mixotrophic, meaning that they can be autotrophic or not, depending on the environmental conditions. So the discovery of this Archaea have changed our view of how the ocean works, and in particular how the deep ocean works, because for a very long time most let's say marine biologists and microbiologists concentrated on the study of the surface layers, the euphotic zone going down up to or down to 200 meters depth, more or less, because it was here where we thought most activities concentrated. Here takes place, photosynthesis takes place, and there is an active cycling of organic matter. And deep sea communities, at least planktonic communities, were thought to rely to have very low metabolic rates and to rely mostly on particulate organic matter coming from the surface, with only very minor input of chemosynthetic carbon coming from deep sea beds. But essentially, planktonic communities depending on the organic matter coming from above. Most dissolved organic matter, the sugars, etc., everything that is easy to crack, is already recycled in the upper parts of the water column. So the discovery of these Archaea that are very abundant in the deep sea has changed this view, because we know now that, okay, heterotrophy is important in the deep sea, but also autotrophy, because up to 1/3 of the biomass, at least the bacterial biomass, can correspond to this kind of organism. So this has also been very active. So these Archaea are very much investigated because of that. So what do we know about the two other groups of Archaea? There are sister groups, group 2 and 3 Archaea, which fall in the Eukaryotes branch. What do we know about them? So they are much more enigmatic. They are exclusively marine. We have no species culture in the laboratory, so it's impossible to study them in culture. Their metabolism and lifestyle is unknown. And in the case of group 2, they are more abundant in surface waters, and particularly at the deep chlorophyll maximum. And the group 3 appears to be really specific, or it has been essentially identified in deep sea waters. Even if we don't know what is their metabolism in general, we have some clues, at least for surface Eukaryotes, because several years ago, Frieden and co-workers identified in metagenomic, in phosphate clones, in genomic clones of the surface waters, a particular gene, here labeled in red, sitting beside the 16S chromosomal RNA gene, and this gene encoded only in organisms coming from surface layers, or relatively surface layers, this is a biphotic zone, a gene encoding a proteorhodopsin. So this is a proton pump that is known to be involved in the generation of a proton gradient when it is activated by light. So this suggested that these organisms, in the surface at least, might be photoheterotrophic, gaining an extra of energy by light activation of this protein, okay, the protein encoded by this gene. And this was further confirmed by the first consensus genome obtained from metagenomic sequences, in this case massive genomic sequences, that was obtained from surface waters by these people, Iverson and co-workers, using, and this genome is just a composite of 5 or 6 different strains, so it is an average genome, and it contains a proteorhodopsin gene, again suggesting that these organisms in surface where they are abundant, they might be photoheterotrophs. Okay, so this example highlights the benefits or the potential use of metagenomics to get access to the genomes of uncultured microorganisms. So when, there are 2 possible ways to go when you do metagenomics. You can extract, what the idea is always the same, you analyze the genomes of a particular environment. So in this case, this would be deep sea water, for instance, that you can collect with this kind of whisking bottles in rosettes, and then you extract the DNA of the fraction that you want, and then you can have 2 possibilities. You can either do massive sequencing. Today this will be done essentially by high throughput techniques like pyrosequencing or Illumina sequencing, and this strategy would give you information about the gene content of a given environment, but it can also provide genome scaffolds of relatively abundant organisms. So you will also get eventually information about genomes that are dominant in your system, because you could eventually assemble part of them. Otherwise, you just have information about the gene content of an environment. The other possibility, which is more expensive but interesting, is to build metagenomic libraries. This means that you clone big fragments of DNA, so big genome fragments, in phosmids. So this allows to clone things between 25 and 40 kbs, so big pieces, or bacterial artificial chromosomes. And in this way, you can sequence those clones, and in this way you can also get information about the gene content of your environment, but you can also have access to the genome structure of organisms that now you can place in a phylogenetic tree, because if you have all the genes in a genome fragment are physically linked, and this means that if you have one gene that can be used as a phylogenetic marker, you can place this genome fragment in a tree and more or less imagine the kind of organism it belongs to. And in this way, you can have access potentially to functions and genome structure of that kind of organism that you can now place in a tree. And this is in a sense the same type of information that now you can also, or people are starting to get also by single-cell genomics. For the moment, single-cell genomics provides only partial sequencing of genomes, and this is more or less the same, or equivalent. So the story that I'm going to tell today started several years ago when we constructed metagenomic libraries from deep sea waters coming from 1,000 and 3,000 meters deep in the Mediterranean or in the South Atlantic here. In phosmids, so genomic clones between 25 and 40 kbs, And these libraries were relatively enriched in Archaea, both Eukaryotes and Archaea. So, in order to characterize the diversity of Archaea that have been captured by our libraries, we looked for 16 ribosomal RNA genes in the plasmids that we got. And we identify a series of OTUs here, both belonging to the Archaea and to the Eukarya, to the different groups, 1, 2, 3. And these OTUs, some of them were really abundant, were cosmopolitan because we have nearly identical sequences from the Mediterranean Sea, from the South Atlantic, and also you have sequences coming from the Pacific, so meaning that these are cosmopolitan OTUs. And one of the things that we observed when analyzing these plasmids that we sequence, so those containing 16 ribosomal RNA genes, we got the full sequences, and we, by just making initial blast analysis of the genes present in these genome fragments, we saw many potential cases of horizontal gene transfer. So we decided to make maximum phylogenetic analysis by maximum likelihood of all the genes present in these 21 plasmids carrying 69 ribosomal RNA genes, but also of the genes that were placed at the end of inserts or genome fragments cloned in our library. And we got many plasmids that had extremities matching Archaea, but many of them, nearly 600, had one extremity typical of Archaea and the other typical of bacteria. So perhaps this could be putative cases of horizontal gene transfer. So we analyzed the trees in detail, but before going on, I want to make sure that you understand the way we interpret phylogenetic trees and the topologies of these trees and how we decided whether something was a horizontal gene transfer or not. If we have a gene that in the different organisms, bacteria, eukaryotes or Archaea, or let's say in just two domains, bacteria and Archaea, form monophyletic groups like that, we are going to conclude that this is a universal gene. And this is vertically inherited, okay? If we see a case like this, with essentially the three domains of life, and well supported, and now a couple of cases like this, we are going to conclude that these are cases of horizontal gene transfer from this lineage to the other two. So this is a gene that is universal, and it has been transferred occasionally to other lines. If we have a case like this, where you have, for instance, all the bacteria, so this is a typical bacterial gene, and all of a sudden you have a group of Archaea in the middle of bacteria, and this is well supported, then this is a case of a domain-specific gene, it would be a bacterial gene, that has been transferred to the Archaea in this particular group. Okay? And then when you have cases like this, with all the branches mixed, or when you have cases with very few genes present, these are cases that we consider unresolved or poor sampling, and then we exclude them from our analysis. We don't analyze these genes, we don't count them. Okay? So applying this type of analysis, from the analysis that we made of all the phylogenetic trees for all the genes in the 21 plasmids, we observed that between 20% to 30% in the case of groups 2 and 3, Eukarya, were cases of horizontal gene transfer coming from bacteria, so very distant donors, another domain. And if we looked at the extremities of genomic fragments that were cloned in our libraries, we detected that between 10 and 20%, depending on the different libraries, corresponding to genome fragments with one extremity of Eukarya type and another bacterial, or between 10 and 34 Eukarya and bacteria. The other case was Archaea and bacteria. So with Archaea bacterial lens, suggestive of potential horizontal gene transfer, but we needed to test it further. are these cases real? So this is the question that I'm asking here. If they are real, are they genome-wide? Is horizontal gene transfer widespread in the genome? Is horizontal gene transfer ancient, recent, or going? And which are the genes? What kind of genes are transferred? So in order to answer to that, we sequence almost 1,000 phosmid clones from these metagenomic libraries belonging to both Tomarchiota and the groups 2 and 3 Eukaryotes. And this represented like 16 Tomarchiota genome equivalents and 9 group 2 and 3 Eukaryote genome equivalents by analyzing or counting the average number of genes, a series of 40 genes, that are usually found in single copy in prokaryotic genomes. Okay? So this also corresponds very well with the number of RNA genes that are present in the that we detected both in the Tomarchiota and Eukaryote. So we of course had looked for the potential functions of these. Well, one of the things that we did was to class the genes in orthologous gene clusters. So these are genes that are orthologs, so homologs, in the two cases, with 2,000 and 3,500 more or less for the Eukaryote. Then we got some functional hints about the type of functions encoded by these archaeal genomes. In the case of Tomarchiota, for instance, we detected genes related to the oxidation of ammonia, like AMO-C or ammonia transporters, and also the metabolism of urea. These genes are in single copy, so you can compare them with other single copy genes in the genome. So this suggests that these organisms are actually ammonia oxidizers because they have the genes for and they have one copy per genome. They have also the potential for autotrophic carbon fixation because we identified all the genes that in Nitrosopuminus maritimus have been described as members of the carbon fixation cycle. And in the case of the group 3 and 2 Eukaryote, we couldn't detect any homolog of proteol or rhodopsin, and this suggests that this group of Eukaryote inhabiting the deep sea are genuine deep sea dwellers. They are not typical of the surface that are, for instance, sedimenting down to the bottom because they are not, they don't carry big genes, so they are not activated by light, and they probably are versatile heterotrophs that can use different kinds of organic matter for energy gain. But what I'm going to talk about today is more on the phylogenetic aspects. So one of the things that we did was to try to reconstruct pangenomes for the two major lineages, groups 2 and 3, Eukaryote and the whole Tomarchiota. So all the sequences here in color corresponds to phosmids that we got and that contain 69 ribosomal RNA genes in our case. And we did that including also, in the case of Tomarchiota, 3 additional reference genomes from databases. And in the case of Eukaryote, the only available genome, that is the genome composite that I mentioned before, obtained by direct metagenomic sequencing. Okay? So we constructed, or we produced lists of gene complements belonging to the pangenome of these two large lineages. Okay? So there's a problem, please tell me. So once we have this list of genes that were, let's say, belonging to that whole lineage, the two whole lineages, we classified them, we distributed them in classes according to their phylogenetic origin. So we defined core genes as genes that were universal or universally present in Archaea. So those we consider to be core genes. We define them lineage-specific genes, core genes, to those genes that were shared by all the Tomarchiota to the exclusion of the rest of Archaea, or to all the Eukaryotes to the exclusion of the rest of Archaea. And finally, we define shell genes as genes that were only shared by a few lineages, one or two, or just one, okay? Or phosmids in our case. And then what we did is to include all the orthologous genes that we had identified for the Tomarchiota, for the Eukaryotes. We add them to our local database that contained several hundreds of genomes from both Archaea, bacteria, and eukaryotes. And then with that we made phylogenetic analysis using maximum likelihood in an automatic mode. And then we looked at these trees manually to decide about the phylogenetic origin of each one of the genes. And with that, we were able to refine also this encircle, let's say. So we refined our classification. So by doing this phylogenetic analysis, we detected cases of genes that have been transferred from, in this case, bacteria to Tomarchiota in the shell class. So this is one example. You have here two Tomarchiota phosmids, or this is particular gene, coming from, in this case, gamma proteobacteria, and you have other proteobacteria around, so this is a clear case. Here you can have another clear case, in this case to group 2 Eukaryotes, to one of the phosmids, which is nested within the gamma proteobacteria with very good support, so this is not a problem. So we consider this as late horizontal gene transfer events to a recent import, because these appear only in a few of our phosmids or genomes. But we also detected horizontally transferred genes in lineage specific core class, in this class. So for instance, here you have a case of a genome that is shared by all the Tomarchiota, including the reference genomes. So it was acquired by the ancestor of the whole group of Tomarchiota coming from bacteria. You have it in the middle of proteobacteria here. And the same happened in the case of group 2 and group 3 Eukaryotes. You have here an example of a horizontal gene transfer case from bacteria again to the ancestor of this group of Eukaryotes, including also the composite genome that is available from the literature. So these cases, we consider them ancient horizontal gene transfer levels because the gene was acquired by the ancestor of the respective groups. Okay? And in the case of Tomarchiota, nearly 70% of these genes were also shared by Nitrososphera gardensis, which is a representative of soil. So these are possibly really ancestral cases that that predate the divergence of soil and marine Tomarchiota. Okay, and if we count all these cases, what we have is that almost 24% of the genes in the Tomarchiota genome are coming from distant horizontal gene transfer, about half for shell genes and the other half for specific core, so early transfer cases. And up to nearly 30% of the genes in group 2 and 3 Eukaryotes genomes are coming from, again, distant horizontal gene transfer donors. No? So in this case, you see that there are more genes belonging to the shell portion than to the specific core, but this is possibly due to the fact that we don't have good reference genomes. The only reference genome that we have is a composite genome that may lack some genes actually. So when we look to the donors of these genes, actually most of them are bacteria, except in the case of the shell class for the Tomarchiota, where up to 20% of these cases are coming actually from Eukaryotes. So it's an inter-phylum transfer. And in the case of bacteria, many of them are not very well identified, so we know they are bacteria, but we cannot pinpoint the exact donor. But when we can do that, most of them are proteobacteria in blue here, or other groups of bacteria that are also frequent in oceans. Proteobacteria are very widespread. So we tried to dig a little bit into this, and we looked for, in particular, signs of recent transfer in the genome. And this is usually done by looking at things like codon usage, because normally genes that had been recently acquired to a new genetic or genomic environment are not adapted. They don't have the codon usage adapted to that of the organism. So we try to look for that kind of signs of horizontal gene transfer. So this is just the principal component analysis of codon usage. In just the Archaea and the Eukarya, you see two clouds here, and this is due to the different GC content bias. So different codon usage will occur in the two types of genomes, because in fact the Archaea have a much lower GC content than the Eukarya, so we can distinguish very well the two clouds, genes belonging to the Archaea and to the Eukarya. But now if you color the genes in the Archaea or in the Eukarya, if you color them according to the class, if they are transfer genes early or late, or if they are core genes, actually you don't see any difference. Both the late and the early transfer gene classes are mixed together. This suggests that even the recent cases of horizontal gene transfer are rather ancient, because the genes have already adapted their codon usage to that of the host. Okay? So, they have already adapted to the codon usage. We also looked at the functional categories of the genes that had been acquired by Archaea from distant donors, from bacteria essentially. And here, so what you see here is for the Archaea and the Eukarya, and the functional classification according to COG, the COG classification system, and to the PEG. So here we are looking to pathways. And in both cases, actually for the different types of genes, the archaeal core, the specific core not affected by horizontal gene transfer, the specific core, the early transfer cases and the late transfer cases, when you look at the relative distribution pattern of genes for the different classes in Archaea and Eukarya, both for COG and for K, you see that there is a significantly, and this is significant actually, similar distribution. So the patterns are more or less the same for these equivalent categories. So of course the category of Archaea core genes are more occupied by information storage and processing genes. Genetic information and processing are more abundant here, and also metabolic genes, etc. But those categories of genes transferred either with early or late transfers are essentially dominated by genes related to metabolism. And this is something that is expected in prokaryotes. Most of horizontal gene transfer affects genes related to metabolism. Another thing that is remarkable here, that maybe, well, it's maybe not that important for you, is that the number of genes that in the specific core for Archaea and Eukarya that have not any known function is remarkable, and this actually highlights how little we know about these groups of Archaea. Well, that's another point. So when we look, you don't have to read the slide, but when we look into more detail into the functions of these genes that are transferred, many of them have to do with the transport across membranes of different things, also energy production and conversion, which is related to membranes, and cell and wall membrane biogenesis. So this is a very important function. And so these functions have to do actually with the membrane, and this suggests that these genes have played a role in the adaptation to a novel environment, because the membrane is the first or the immediate contact with the surrounding environment. So this is something fit for thought. So this is our observations in these particular two groups of non-uncultured archaea for the deep sea. But interestingly, a couple of years ago, in 2012, Nelson Salti and co-workers made a similar observation in another group of archaea, the haloarchaea, called traditionally halobacteria, because before we knew they were archaea. And actually, they showed that the ancestor of haloarchaea, which was a methanogenic archaea, had acquired massively, so up to 1,000 roughly genes, also from bacteria, and they said that these genes were very important in facilitating the passage from an anaerobic metabolism, methanogens anaerobic, to an anaerobic respiration mode, which is that use the haloarchaea. So this is interesting because when we combine our two observations, in our case, when looking at the different trees, we, in many cases, in several cases, we observe genes that were transferred from bacteria not only to the Tomarchaeota and to the group 2 and 3 Eukaryotes, but we also observe haloarchaea being placed in the middle of bacteria, so suggesting an independent transfer of the same gene to the three groups independently. They are the same bacterial gene, but coming from different groups of bacteria to the three archaeal lineages. And we have also detected cases like this one, where you have two independent acquisitions from bacteria of the same gene by one group of archaea, and either, in this case, halophiles or Tomarchaeota, and then internal transfer within archaea. Okay? And here you have the opposite case, with again, one transfer to, in this case, haloarchaea, and either to Eukaryotes or Tomarchaeota, and transfer between the two groups. Okay? So when you count the number of these genes that are shared, actually you have that up to 1/3 of the genes that have been acquired by Tomarchaeota from bacteria are also shared by halophiles, and up to 1/4 of the genes that have been acquired by group 2 and 3 Eukaryotes from bacteria are shared with haloarchaea. Okay? So here you have the numbers. So, and this is my last slide, so you don't have to... no, this is not the last one. So when we look into the functions of those genes, actually most of them correspond, I summarize, to energy metabolism, membranes and lipids, and again, amino acid transfer and metabolism. So in many cases, genes related to the membrane and to the energy gaining. So many of them are genes like cytochrome oxidases related to aerobic respiration, but also many genes that have to do with membranes. So membranes are very important in the adaptation to lower temperatures because you have to adjust the flexibility of the membrane when you move from temperature. So it is very tempting to speculate, and this is my last slide, that in the same way, and now we are looking at the tree of life if you want, the eukaryotes are not here, but the bacteria and the archaea, it is tempting to suggest that in the same way as we know that some hyperthermophilic bacterial groups have adapted secondarily to hyperthermophily by acquiring some genes from hyperthermophilic archaea, such as the reverse Chilis specific enzyme for hyperthermophiles, it is likely, or this is the hypothesis that we would like to put forward, that the reverse is true for archaea, and that archaea independently adapted secondarily to mesophily from a hyperthermophilic ancestor by gaining genes from bacteria that favor the adaptation or facilitated the adaptation to a series of lifestyles that are more frequent in low temperature environments or in mesophilic environments in general. And with that, this is just a summary of what I say. With that, I just thank you for your attention, and these are the colleagues that were involved in this work. Thank you very much.
Yes. have time for questions.
I know this is a bit far from maybe your background, but I guess this is good to know.
What do you think? Is it possible to imagine a horizontal gene transfer of 60 S ribosomal RNA gene?
It would be possible if you have good examples for that. It can be transferred, so there are a few examples that can happen. It is not massive. So I think that it may be frequent, but then it is difficult to measure between very closely related organisms because you don't disturb the ribosome. Okay? If you import a gene, 16F ribosomal RNA gene from something that is really different, you're going to have problems with your ribosome, and the ribosomes are very important for you. So you don't want that. But it may be likely, why not, that you can gain or exchange with close strains, and that it's very difficult to measure. You don't know. In some cases, that exists. For instance, it has been shown in some cases where you can have an adaptation for resistance to antibiotics, etc., that can favor that type of thing. There are few cases described, but I don't think that this is massive for this type of genes. And actually, in this particular case, I don't think it is a problem because we don't only have one gene that is the 16F ribosomal RNA gene, we have the whole genome complement, and there are many Archaea genes and proteins that are really constant and that are domain specific. So I think there is good evidence in this case to say that this is not the case. But there are also cases in some Archaea you have two different genes that can differ up to 5%, so that's a significant difference. And in those cases, people have shown that they can be expressed, the two different genes, under different conditions. So they may, they are adaptations to different local conditions.
Okay, thank you.
Is it true that humans acquired some genes from Neanderthals?
From? I didn't hear.
I don't really know English. Neanderthals.
Well, that's another story. That is hybridization. Okay. I think, well, that could be perhaps a form of horizontal gene transfer, but that has to do with sex. So populations met and interbreed. So that's a slightly different story. So we are hybrids in some way, yes. But that kind of recombination in eukaryotes is encoded. You have sex, so you mate two different organisms, and you have a specifically guided recombination between paired chromosomes. So that's another way of generating actually genetic diversity. That's what we want to do to avoid problems. But that's the way it happened. It is different.
And you've mentioned a lot of times, if I'm not mistaken, that the major donor group of those genes that are horizontally transferred to Archaea is the proteobacteria, yeah?
In many cases, in cases where we can identify the donor, but in most cases, we cannot say which bacterial phylum.
Yeah, okay. Can you give maybe your explanation to this fact? Is this just a mere coincidence, or are these genes certainly very useful and so they are transferred, or do proteobacteria have some physiological adaptation to transfer? Just very large group.
Proteobacteria are extremely diverse and abundant in nearly every environment. So if you go to soils, if you go to the sea, they dominate there. They are also extremely varied in terms of metabolism. You have autotrophs, you have heterotrophs, so you have a wide variety of metabolisms. So possibly some of the genes were interested But they are also the most likely donors because they are the most abundant. So I can explain.
Okay.
So shall we have a break and continue?
Okay.
Oh, sorry.
Did you try to estimate the time when these cases of horizontal gene transfer occur?
No, we would like to, but it's a very difficult thing to do. Yeah, no, we don't have that, we don't know.
Okay.
There is not a good molecular clock for these organisms, but it would be interesting.
Do you see any other questions? Yep.
Break.
Then it's a break time.
Okay.
Thanks.